Good morning, students. I am Ms. Rupablika Borgohain, Assistant Professor, Assam Downtown University, Guwahati, Department of Pharmaceutical Science. So now I'll be discussing about the qualitative test of carbohydrate, that is Barfoed test. So the principle for Barfoed test is aldoses and ketosis can reduce cupric ions even in acidic conditions. This test is used to distinguish reducing monosaccharides from disaccharides by controlling pH, and the time of heating is also maintained. Monosaccharides react very fast, whereas disaccharides react very slowly. So now comes the procedure for Barfoed's test. First of all, we have to take 2 ml of Barfoed's reagent, add 2 ml of carbohydrate solution to it. Keep the test tubes in the boiling water bath for 3 minutes, then cool under running water. Overheating should be avoided. So the interpretation is the positive reaction indicates the presence of a reducing monosaccharide. On prolonged heating, disaccharides can also give this positive test. Hence, the solution should be boiled for 3 minutes only. Now comes the Selin-Windhorst test. So the principle for Selin-Windhorst test is ketohexosis on treatment with hydrochloric acid form 5-hydroxymethyl-furfuran, which on condensation with resorcinol gives a cherry red colored complex. So the procedure for Selin-Windhorst test is we'll have to take 3 ml of Selin-Windhorst reagent and 1 ml of fructose to it. Then boil it for 30 seconds only. Cool the solution. Then the interpretation comes, this test is given positive by ketohexosis, so it is answered by fructose, sucrose and other fructose containing carbohydrates. This test distinguishes between glucose and fructose. Overheating of the solution should be avoided. Upon continuous boiling, aldoses get converted to ketosis and give a positive reaction with Selin-Windhorst reagent. Now comes the hydrolysis test for sucrose. So the principle is sucrose on hydrolysis with hydrochloric acid is converted to glucose and fructose. The presence of these two monosaccharides can be confirmed by Benedict's and Selin-Windhorst test. So the procedure is we'll have to add 2 drops of hydrochloric acid and 1 drop of thymol blue to 5 ml of sucrose solution. Then there will be the development of pink color, which indicates that a solution is acidic. Then divide it into 2 equal parts. Then one portion is boiled for 1 minute and the other portion is kept for cooling under tap water. Then neutralize both the portions by adding 2% sodium carbonate drop by drop. Then we'll see there is a formation of blue color which indicates neutralization. So Benedict's and Selin-Windhorst test with the boiled portion are performed. Now the interpretation is sucrose is a non-reducing sugar since it doesn't have free aldehyde or ketone group to cause reduction. Hence it gives a negative reaction with Benedict's reagent. But upon boiling with hydrochloric acid, sucrose is hydrolyzed to yield glucose and fructose, which give positive reactions with Benedict and Selin-Windhorst reagent. Now discussing about the Osazon test. So the principle is a solution of reducing sugar when heated with phenyl hydrazine, characteristic yellow crystalline compounds called Osazone are formed. These crystals have definite crystalline structure, precipitation time and melting point for different reducing sugars. Now the procedure for Osazone test is we will have to add 10 drops of glacial acetic acid to 5 ml of sugar solution in a test tube. Then we'll have to add a knife point of phenyl hydrazine hydrochloride and double the amount of sodium acetate crystals. Then mix and warm a little to see that the solids are dissolved. After that, the solution is filtered in another test tube and keep the fill rate for boiling up to 20 minutes. Then allow the tube to cool slowly in the water bath without cooling it hurriedly under tap to have beta crystals. So under the water bath, we'll have to keep to cool for slowly so that beta crystals are formed. Then after that, we'll have to examine the crystals under the microscope. So these are the Osazone test. After completion, we'll get the crystals in these forms. So finally, glucose, fructose and mannose produce the same Osazone because of the similarities in their molecular structure. Galactosazone crystals are formed in 7 minutes. Maltosazone crystals are formed in 10 to 15 minutes. Now comes the Biles test. The principle for Bayes test is the test reagent dehydrates pentoses to form furfural. Furfural further reacts with ursinol and the iron ion present in the test reagent to produce a bluish product. So the procedure for Bayes test is we'll have to take 2 ml of a sample solution which is placed in a test tube. Then 2 ml of Bayes reagent, a solution of resveratrol, HCl and ferric chloride which constitutes the bile reagent is added. The solution is then heated gently in a Bunsen burner or hot water bath. The interpretation is this test is specific for pentoses. Hexoses generally react to form green, red or brown products. Now comes the iodine reaction. So this is a test for polysaccharides. The principle for iodine reaction is iodine forms a coordinate complex between the helically coiled polysaccharide chain and iodine centrally located within the helix due to adsorption. The color obtained depends upon the length of the unbranched or linear chain available for complex formation. Interpretation. Amylose, a linear chain component of starch, gives a deep blue color. Amylopectin, a branched chain component of starch, gives a purple color. In case of glycogen, it gives a reddish brown color, and in case of dextrins, amylo, erythro and ectrodextrins formed as intermediates during hydrolysis of starch gives violet, red and no color with iodine respectively. So these are the reactions of glucose. So in case of Mollis test, the observation is purple ring at the junction of two liquids. So inference we get fructose is a carbohydrate. In case of Benedict's test, brick red PPT and it depends upon the amount of sugar present. So the inference is fructose is a reducing carbohydrate. In case of Burfoyt's test, scanty red PPT at the bottom of the test tube occurs and the inference is fructose is a reducing monosaccharide. In case of Selivinov's test, cherry red color is observed where fructose is a ketohexose. Then lastly, in case of Osazan test, needle-shaped crystals are formed where we can say that fructose, glucose and mannose form similar crystals. So in my next video, I will be continuing with the next part of the test to identify the unknown sample of carbohydrate. Thank you.